Hello, everyone. Let's have a little lesson on what the meaning of some words. This is from Patrick Jordan's book, Prion Agenda. Um, I'll put the links down to Lulu. You can get actually a 10% discount right now. But let's see what this says. These words are the trans words, okay? Transcription. Instructions from DNA are copied by tRNA that is then shuttled to the endoplasmic reticulum to build proteins. Transcription is the translation by transfer RNA. Whoever the redheaded stepchild is that developed this naming structure needs to be beaten in public. Transduce, the transfer of genetic material from one bacterial cell to another cell, a process often accomplished by bacteriophage that injects chunks of bacterial chromosomes but no viral DNA, or a plasmid, which is a circular, highly infectious, transducible DNA. Transfect, as per my schizophrenic dictionary, is to infect a cell with bacteriophage, animal, or plant virus so the infected cell replicates a complete virus. But the multiple industrial sources that do all this shit for money indicate that it's primary a non-viral method of getting nucleic material into a cell by using things like plasmids or lipids. Liposomal anything ought to make your testicle crawl. And I talked about the lipid nanoparticle vesicles or delivery systems for the short interfering RNA and the messenger RNA in my last video. Reagent created nanoparticles to chaperone plasmids, microinjection, and electroporation. Still want to do a Bob Beck's electroporation pulsar? I'm not sure what that is. I'll have to look that up. Bob Beck's electroporation pulsar. Since these folks take your money, they don't care about the dictionary definition. If you want viral RNA stuck up the ass of a cell, then by God, that's what you will get. Lay your money down. Transient transfection is a process that gets plasmids and expressible genes into a cell without incorporating it into the host chromosome. Polyethylenamine, PEI, and calcium phosphate are used as vehicles to get the shit inside the cells. I would read doof labels from now on to make sure that you aren't facilitating your own rewrites by eating prepared meals. Stable transfection gets the nasty bits installed in the cell as an episome or inserted into the host chromosome, like putting a cat inside a mailbox. That used to be a prank out in the country in the grain ghetto. Believe me, you don't want to be the one opening that box. In order to make sure that the cat got to the right address, they used markers, on one of which being dihydrofolate reductase. Now, some of these words are just difficult to pronounce. That alone should make your whiskers tweak, but if it doesn't, then DHFR might get you closer to MTHFR so that you see what they're doing in the lab is indistinguishable from what they're doing on the street. Oddly, a corporate specializing in transfection procedures says that stable transfection is rare. Transform, to genetically alter a cell with free, naked, foreign DNA from another cell or a virus. This is the essence of what everyone fears from GMOs. To put the gene for human insulin in bacteria so that it overproduces that industrial product for harvest, converting normal cells into an immortal line of cancer cells, is called transformation. The HeLa cells from Henrietta Lacks' cervical cancer can infect, transfect, transform whatever they come in contact with to become the undead. Now, yeah, I did a video where I talked about the HeLa cells. So, transformation. When an oncoretrovirus inserts itself into the host genome, it converts that cell into a cancer. A cancer is a new species. It's a parasite that replicates uncontrollably at the expense of the life of the host. When a cell is transformed, it's infected, converted, and cancerous. It is then said to be immortal. It will continue to grow and reproduce as long as there is a food supply. The nutrient requirements for normal human cells is complex. Cancer has a minimum food needs. Cancer cells have minimal food needs. Translation. DNA is a code. RNA is a mirror of that code that is read at the endoplasmic reticulum that translates the code into a series of amino acids that are strung together to make proteins. Let's say that again. DNA is a code. RNA is a mirror of that code, and that is read at the endoplasmic reticulum that translates the code into a series of amino acids that are strung together to make proteins. So, in what we were learning yesterday, they are trying to stop translation and rewrite. So, musical chair of genes. Translocation is a musical chairs of genes. This can occur in the host genome or under the influence of an infectious agent. So there you go. There we have it. No, we got more. Transvect. Okay. Coined in the genetic sense by Edward B. Lewis in 1954, an epigenetic interaction between allele on one chromosome and the corresponding allele on the homologous chromosome. Transvection effects include both activation and silencing. The act of supernatural flight, such as BioWitch 1996, Abner Falk, a psychoanalytic history of the Jews, ISBN 083-863-6608, page 532. In Transvection Witches, rode broomsticks, a classical phallic symbol. Definitely witchcraft going on here. Transvection. Insertion of genetic material by a bacteriophage virus. This can be the phage inserting its own material into bacteria to take over the cell's function, or it can be an artificial manipulation of a phage to carry genetic material that was put into it by the military. Transvection was a process that revolutionized biology and biophysics to become molecular biology. It was the beginning of genetic modification. The classic T phages can be thought of as syringes that insert genes into a host using restriction enzymes to insert those gene sequences into the host nucleus, and then using the host machinery to make copies of itself. Once this process was understood, then all life was reduced to an engineering problem that could be solved by science. tRNA. So the tRNA is the same as messenger RNA? Messenger is the copy of DNA that gets taken to the endoplasmic reticulum to be decoded and translated. A confusing damn word starting with T, into functional molecules, with transfer RNA shuttling amino acids to the rRNA in the ribosome factory for assembly. Another popular option is the use of transfection reagent. Several companies offer specialized short interfering RNA delivery reagents. Please consult the transfection reagent vendor's protocol for detailed information for the exact requirements and procedure. Careful optimization of variable factors should be ensured for all initial transfection experiment. It is based on this and further optimization that reproducible gene knockout results will be obtained. Usually, RNA interference effect is seen within 4 hours, and the maximum downregulation observed in 24 to 48 hours. The effect lasts several cell generations and from 4 to 10 days, depending on cell culture. Wow. So, how about that lesson in trans? And again, remember that you can get Patrick's books on Lulu. And there we have the Prion Agenda. They're offering 10% off right now. Thanks for watching, guys. Let me know what you think.